transport of CO2. See, as we all know that whenever we inhale, then we are inhaling majorly oxygen, and whenever we exhale, so majorly we are exhaling the CO2. Okay, but now how these gases, now oxygen transport we have already seen. Now we are majorly focusing on the transport of CO2. How exactly the CO2 will be transported in the blood? Okay, and how we will be removing the CO2, that we will see in this topic. Okay, now we might have seen initially only that oxygen 97% of oxygen is going in the form of HbO2, okay, via RBC. Yes, with hemoglobin 97% of oxygen is going. Only 3% of oxygen is going in dissolved state. But here you will see that maximum 7% of the CO2 will be going in the form of dissolved state. Let's say in plasma it will be going in the dissolved state. Why? Because this CO2 is 20 to 25 times more soluble. This CO2 gas is more soluble compared to the oxygen. So that is what we need to see here. So in this case of CO2, here we have 23% of the CO2 will be, in NCERT it is between 20 to 25 on an average we can take 23% of the CO2 will be going in the form of HbCO2, that is known as amino hemoglobin. It means 23% of the CO2 will be transported in the blood in the form of HbCO2, that is known as amino hemoglobin. Now, why we are not telling carboxyhemoglobin? Because carboxyhemoglobin is a different thing. When carbon monoxide will bind with the hemoglobin, that is what the term carboxyhemoglobin. Carbamino hemoglobin, why we are using the word amino? See, we all know hemoglobin is a complex protein, okay? And in hemoglobin, globin proteins are there. Basically, globin chains are there. So this particular CO2 will be binding with the end terminal of globin chain. That is why the word we are using is amino, okay? Carbamino. Why? Because CO2 binds to end terminal of globin chain. So this is 23% we are going, 23% of the CO2 is going in the form of HbCO2. Now 7% will be going in dissolved state or in dissolved form to plasma. And then rest 70% will be going in the form of bicarbonate ions. This bicarbonate ions is also transported via plasma only. Okay, now if you see this 23% which is going in the form of HbCO2, now this HbCO2 we all know hemoglobin is in the RBC, so this you can say through RBC. So 23% is going through RBC, and this nearly 77%. This bicarbonate is also transported via through plasma. So total 77% is going. Total 77% is going via plasma, and 23% is going via RBC. But there is a very important thing now. As I have already told you that maximum amount of CO2, 70% is going in the form of bicarbonate ions. But there is very important thing. This bicarbonate ions is made. This is made in RBC, but transported via plasma. Now how we are making, why we are making in RBC? Because see, there is one important enzyme that is known as carbonic anhydrase. Carbonic anhydrase is actually responsible to make these bicarbonate ions. Okay, so that is why the making is done in RBC. Once you make the bicarbonate ion, then it will be transported in the flow. Clear? So carbonic anhydrase is the enzyme which is found abundant in RBC. A little concentration you can find in plasma also. Okay. Okay. So we will go with the process now. Now a rough fraction I already give you how much percent it is going. 23% it is going in the form of HbCO2, that is carbamino hemoglobin. Then 7% it will go in the dissolved state, and 70% it is going in the bicarbonate ions, you can say, in the form of bicarbonate ions. Okay. Now we have to see the loading and unloading. Where you will load the CO2. See. In short, we know that when you break the glucose we are breaking at the cellular level or the tissue level. Okay, when we break the glucose, then you can get what? Finally, you can get ATP, you can get CO2, and you can get water. So we are breaking the glucose oxidation is done at cellular level or tissue level. Okay, so maximum CO2 you can get at tissue level. So from there we have to take the CO2 and then we have to release the CO2 near to the lungs, and then from the lungs you can easily exhale the CO2. Okay. Now how you will bring that CO2 from the tissue to the lungs, that is what we need to see. So loading and unloading of the CO2 we have to see. Now for example, we are going with the loading. The first step is loading. Loading of CO2. Okay. Now this loading of CO2 happens where? So I have already told you, tissue level. At tissue level you will see this thing. Okay. Now we all know one thing clearly, that at tissue level the partial pressure of oxygen is less and the CO2 is more. You have to keep this in your mind that at tissue level your PCO2 is what? It is more and your P oxygen is less. It means partial pressure of oxygen is less at the tissue level. Okay. Now we'll see. For example, if I make this is the tissue for us, and then we will leave this is the RBC for us. Okay, this is what the RBC I am drawing. Maybe here we will have the blood plasma. I know. Okay, this is the tissue for us. This is what? This is the tissue. This is the RBC. Now in tissue we all know that we have our glucose C6H12O6. It will break. You will break the glucose in the presence of what? In the presence of oxygen. Yes, glucose oxidation we are doing at the tissue level. And finally what we will get? We will get ATP plus H2O plus CO2. So the CO2 which we are getting, that is via the breakdown of glucose. So now how you will take this CO2? Now the loading you have to do. You have to take this CO2. Okay. So now as we already know that 7% is going in the dissolved state. It is going in the dissolved form, so that is not the issue now. So we will just, this is going in the dissolved form first, so that is not the issue. Now here we know that here you might have what? HbO2 first. Okay, HbO2 is coming from the lungs. That is, this is oxyhemoglobin. Now the condition which we have at the tissue level is what? Partial pressure of oxygen is less and CO2 is more. In this condition, this HbO2 association will break. You can say the HbO2 oxyhemoglobin will dissociate. Why? Because this is not a favorable condition for the binding. Okay, where you have maximum partial pressure of CO2, this will split. This will split. This will split. Okay, so finally you can get what? Free hemoglobin plus oxygen. Now you can say, now once your hemoglobin is free, this CO2, how much percent? So you can say 23% of the CO2 now will come and it will bind with what? This free hemoglobin, and then it will make what? HbCO2, that is carbamino hemoglobin. Okay, so it means you are breaking the HbO2 and then this free hemoglobin will bind with the 23% of CO2 and then it will make what? HbCO2, that is known as carbamino hemoglobin. So now we are done with this. We are done with the 7%. Now what about the 70%? What about the 70%? This is the 70%. Okay, 70% of the CO2. Now it will be combined. It will be reacting with the water, CO2 plus H2, and then you will get what? H2CO2, means carbonic acid will be. This CO2 will combine with water in the presence of which enzyme? In the presence of carbonic anhydrase. Okay, and then it will make carbonic acid. And this is a reversible reaction, you can say. And then further, this carbonic acid will split in CO2 minus, that is bicarbonate ions plus H+. Okay, this is a very, very important reaction which happens in RBC and which leads to the production of bicarbonate ions. Both the steps are catalyzed by an enzyme, that is carbonic anhydrase. Very important point, student. Carbonic anhydrase is responsible to make bicarbonate ions and it is found abundant in RBC. Little amount of carbonic anhydrase you can find in plasma also. Now once you make your bicarbonate ions, that is what I told you. The making of bicarbonate will be done in RBC. So once you make the bicarbonate, then it will be going outside. So for going outside, if you want to take this bicarbonate outside in the plasma, so you want, there is a channel from where it will go out. Now your bicarbonate is going outside. Yeah. Now bicarbonate, when it will go outside, it will be combining. It will be combining with sodium to make what? Sodium bicarbonate. So once your bicarbonate ion will go outside, it will react, it will combine with the sodium, and then it will be making sodium bicarbonate. Okay. And then one important thing you have to see here, this bicarbonate is a negative ion for you. Okay. Now maximum bicarbonate negative ions you are taking away from the RBC, outside from the RBC, it means what? You are disturbing, indirectly you are disturbing the ionic gradient. Yes. See, if you have a cell and in that cell there is you can say there is a certain ionic gradient, there is certain ionic concentrations, and if you are taking all the negative ions, bicarbonate, all maximum negative ions outside the RBC, it means what? Ionic gradient can be disturbed. Okay, so it means here negative ions will be less inside the RBC. So to cope up this problem, to make the equilibrium, we have to put some chlorine. We will send some chlorine inside. Okay, so we are sending what? We are sending chlorine to maintain the ionic gradient. Clear? So to maintain the ionic concentration, you are just sending the chlorine inside and you are taking out the bicarbonate. Okay, so this gate, if you can see properly, the gate which I am drawing here, this gate from where you are exchanging the HCO3 minus and Cl, this gate has a name, and the name is bicarbonate chlorine exchanger. This is bicarbonate HCO3 minus Cl exchanger, or band 3 protein also is band 3 protein. Clear? So this is what we have seen. This is the exchanger from where you can exchange the HCO3 minus and Cl. Now why we are putting the Cl? Just to maintain the ionic concentration. Okay. Now this particular thing, when we are putting the Cl inside the RBC, this is known as what? Chloride shift. This is known as chloride shift. This is known as chloride shift, or you can say hamburger phenomenon also we can say. Either you can say chloride shift or you can say hamburger phenomenon. Okay. What is hamburger phenomenon? That the chloride ions will be entering the RBC. Okay. Because why? Because we need to maintain the ionic concentration. Clear? Okay. Now one last thing. Now here we have H+. This is the important point for you now. If free H+ will be there, it means what? It will be leading to the acidic condition. And if it is making acidic condition, that will be making harm for the RBC. Clear? So if you have any acidic condition, that will be damaging the RBC now. So we have to suppress this particular action. We have to suppress the concentration of H+. So here, as we all know that NiO2 will break, so you will have a free hemoglobin. Now this hemoglobin will act as a buffer. How? Because it will suppress the action of H+ ion. So it will combine with H+ ion, and then it will make what? HHB. And what is this thing known as? This is known as hemoglobinic acid. Okay. So here very important point is that hemoglobin is acting as a buffer. Hemoglobin is acting as a buffer at tissue level. So why? Because it is suppressing the action of H+. Okay, it will combine with H+ and it will make hemoglobinic acid, that is known as HHB. This is your loading. This is loading of CO2, and which happens at the tissue level, and it is also known as what? It is also known as Bohr's effect. Bohr's effect. Okay, this is known as Bohr's effect, which is inside the tissue level. Now this is the loading. You can see this is the loading. You can see properly. CO2 23% it is going in the form of HbCO2, that is carbonyl hemoglobin. 7% it is going in the plasma in dissolved state, and 70% it is going in the form of bicarbonate. Clear? This is what the loading. Now we'll see the unloading, and after unloading, that is the last one. So nothing happens special in the loading. Whatever we are doing in the loading, just opposite to that is nothing but your unloading. Clear? Whatever we do in the loading process, if you do exactly opposite, that is nothing but unloading. Now we all know that loading we have at the tissue level. From the tissue level, you are taking all the CO2. 100% CO2 we have already taken. Now from the tissue, you have to bring to the lungs level. Okay. Now here we have to see with respect to alveoli. For example, this is the alveoli, and here we have in the RBC. Here we have the RBC. Now what we need to see here? This is the You can see this is the main RBC. This is the condition near the lungs. So near the lungs, what we have the condition? So we have the condition that is partial pressure of oxygen is more here and partial pressure of CO2 is less. Remember one thing. If we have more partial pressure of oxygen, it means this condition will support the formation of oxyhemoglobin. Hb and O2 can make a bond. Okay, they will not detach; they will make a bond. So in this case, whatever the things we have, see what the things will be having. Now HbCO2 we were making at the tissue level. That will be in this condition that HbCO2 will break. Why? Because this condition is not favorable for what? HbCO2. Okay. So now here we will have what? Free hemoglobin plus we will have free CO2. Now this how much percent we were breaking? So we all know this is what 23%. 23% of your CO2 can easily go now because this carbohydrate will detach because of this condition which is present at the lungs level. Okay. Now what else? What else we have? Also okay. So we have this thing. We were having NaCO3 here. Yes. NaCO3 was in the plasma. Now it will again break. It will break into Na plus HCO3 minus. Okay. Then here we will have that exchanger. That HCO3 minus will enter inside the RBC. HCO3 minus will enter inside the RBC. What is the next step now? And when you are taking now the negative ions inside, see initially at the tissue level you were removing the bicarbonate. Now we are taking the bicarbonate. It means we are taking more negative ions. So in this condition, we have to remove the excess of chlorine. Because if you want to take negative ions, you have to remove some negative ions also to maintain the equilibrium. Okay, whatever the chlorine you were taking at the tissue level, that chlorine we will remove here. And this particular, the channel is same. The channel is bicarbonate chlorine exchanger. Okay, but this shift where you will remove the chlorine outside the RBC is known as what? Reverse chloride shift. This is known as reverse chloride shift. Okay, this is known as reverse chloride shift. Once your bicarbonate is now here, okay, we were having one more thing. We were having HHB also. Now this particular thing will also dissociate, and then we will have Hb plus H plus plus. Now here you can see that again we have a separate H plus. A separate H plus we have. Again this condition can lead to the acidic thing, and this can be, you can say this can be giving a damage to the RBC. So here the bicarbonate ions will be combining with the H plus. It means it will react with H plus and it will give what? H2CO3. And here we all know the enzyme will be carbonic anhydrous. And then this H2CO3 will further make what? H2O plus CO2. Okay. And here again the enzyme is what? Carbonic anhydrous. The reaction which we have done at the tissue level, this we are reversing the reaction, nothing else. Okay. This HHB will break into Hb plus H plus, and then further this H plus will be combining with HCO3 minus, which is coming from outside. It will make what? Carbonic acid in the presence of carbonic anhydrous. Then this carbonic acid will split into H2O plus CO2. How much CO2? How much percent CO2 you are bringing in the form of bicarbonate? So your answer should be 70%. So this 70% is now again going away. And then 7% can easily come. 7% was easily in the form of what? Dissolved state. It was already dissolved in the plasma. 7% is coming like this, 70% is coming like this, and then 23% again we are coming like, we are bringing to the alveoli. From here you can remove. From here you can remove the CO2. You can easily exhale from the lungs. You can easily exhale out of the body. Clear now? Now this is what we know. Acha. Here if you see HCO3 minus is suppressing the action of H plus ion. So here at the lungs level, HCO3 minus, that is bicarbonate, is acting as a buffer. Here the buffer, like a buffer, this bicarbonate is acting because it is suppressing the action of H plus ion. Now this all stuff is happening. This condition, if you can see properly, that is partial pressure of oxygen is more and CO2 is less. This condition, this shift is known as Haldin's. This is known as Haldin's effect. Haldin's effect is happening near the lungs, and it will promote the binding of oxyhemoglobin. Okay, it will promote the binding of oxyhemoglobin, and it will detach. It will not promote the binding of HbCO2. This is totally opposite of the Bohr's effect. Bohr's effect was happening at the tissue level. Haldin's effect is happening at the lungs level. This is what we know, student. This is what is known as unloading of CO2. Clear? Okay. Thank you.